Hi everyone, it's Mr. Sinti, and today I'd like to talk to you a little bit about our immune system. Yeah, our immune system is most remarkable. I mean, it protects us from all of these germs that are trying to invade us. And the interesting thing about it is, uh, this particular video is on a topic called cell mediated immunity. So you're going to like this. So our own white blood cells, as you can see in this picture here, all of our blood cells come from bone marrow. And so, are you ready for this? Some of our white blood cells actually destroy our own body cells that are being invaded. That's right. And so, this is what we call cell mediated immunity. And so, let's get into that conversation right now. And so, one of the main characters, we have 5 different kinds of white blood cells which are responsible for defending ourselves. A little shout out to the red blood cells. The red blood cells help to carry oxygen and a little bit of carbon dioxide, so those are pretty crucial. And our thrombocytes, our platelets down there, are important for clotting the blood. But this particular conversation, since it's on the immune system, has to do with our white blood cells. But the king of the white blood cells are lymphocytes. And so lymphocytes develop inside the red marrow, which is mainly found in the epiphyse regions. In other words, the large area of long bones. And so what happens is that those lymphocytes help us to determine the difference between our own cells and non-cells. But what's curious about that is that sometimes they actually go after self cells. And so this is part of our discussion. And so I thought I'd show you this right here. This is technically yellow marrow right here. This is in the diaphysis of a bone, and it's mainly adipose tissue. But I just wanted to show you marrow there, just to so you can get a sense of it. And so what I was mentioning about lymphocytes is that there's 2 main groupings, 2 main types of lymphocytes. They're referred to as T cells and B cells, and those have to do with where they develop and mature, because all the cells are going to be constructed in the bone marrow, the red bone marrow. But where they later develop, if they develop in the thymus gland, which is right above the heart, they're called T for thymus. And if they mature in the bone, they're referred to as B cells. But that be as it may, they have different functions, and so that's what we want to discuss. And so T cells mainly function in cell-mediated immunity, and B are in what we call humoral immunity, which means that they secrete antibodies into the humors or fluids of the body. But what I wanted to say is that when T cells and B cells are maturing in their various locations in the bone and the thymus, they're actually tested, can you believe this, before they leave those locations to make sure that they're competent. And so you don't want rogue white blood cells like lymphocytes to be attacking your own tissue. That would be catastrophic. And so, believe it or not, there actually are some instances where our own white blood cells will attack our own healthy tissue. Like, for example, in what is called rheumatoid arthritis. This is what is categorized as an autoimmune disease. So it's disease because it's a problem. It's not caused by a pathogen, like a disease-causing agent, but it's our own white blood cells that are attacking the cartilage, for example, in the joints, the hyaline articular cartilage between bones. And so this results in arthritis, and this could be a problem. And so our own white blood cells can sometimes confuse self with non-self. And so they're often tested. This is a kind of a cool concept if you think about it, and how intricate is this, where our white blood cells are tested before they're able to go out into the body and defend. And so where are they going out? Well, obviously the white blood cells circulate in the circulatory system, so they defend our body in the blood. But they can also defend our body in the tissue fluid as well, because germs are trying to get out and get us in all different locations. And then clearly, germs then flow from the capillaries into the lymphatic capillaries. And so we have bacteria that are found in the lymph. And so white blood cells are capable of leaving the circulatory system, and they enter into the lymphatic system, And ultimately, they congregate in lymph nodes, and the lymphocytes do most of their damage to germs in lymph nodes. And this is only showing one, but there's many lymph nodes in a series in the lymphatic system. And then once the lymph is cleansed, then that goes back into the circulatory system. It returns. So as I mentioned before, there's two main approaches to this. And this particular video is a discussion of cell-mediated response. But I just want to give a quick shout out to humoral response. And so the B lymphocyte is capable of secreting antibodies. And so let me animate that. And so a B cell secretes these Y-shaped proteins that circulate in the plasma. And they are able to go out and destroy foreign organisms, like for example a foreign germ like a bacteria. And they do that a variety of different ways. And so I point you into the direction of a video that will discuss this on antibodies and how they work in the humoral response. But I just wanted to point that out. Humoral response is antibody secretion, or sometimes they're known as immunoglobulins, these Y-shaped proteins that circulate in the fluids and destroy foreign organisms. So there's two main types. There's the humoral response with antibodies, and the cell-mediated response, which is mainly responsible for destroying our own cells, and our T cells are responsible for cell-mediated. But again, humoral response is when B cells, B lymphocytes, secrete antibodies into the plasma and into the lymph, and they rain down terror on germs. And so here's a B cell, and so what's happening is it's secreting antibodies into the fluids, so it's humoral, and they're going out and destroying foreign cells like that. And they do it a couple of different ways. They can actually cause them to clump together or neutralize them so that they don't actually infect other cells, or make them a little bit easier to precipitate out. But nevertheless, maybe even a neutrophil will come along like this, and I'll animate it, and come along and phagocytize the cells. And so that's what antibodies are capable of doing, is ultimately destroying organisms. And B cells can secrete them. And when they're activated, they divide and they form large numbers. And so the antibodies are produced in quite a large amount. And so that's pretty awesome. And so these circulating antibodies really prevent free troubles. Like, in other words, free troubles, free organisms that are causing trouble, like free bacteria or toxins or viruses in the fluids. Now, and you're like, free? What do you mean? Like, I don't mean in terms of cost. I mean like not in a cell. And so this is what I mean by cell-mediated immunity. Sometimes our own cells, and I'll draw it like this, our own cells will be invaded by germs, like a virus. And so sometimes these germs, like this, here's a virus, and it'll penetrate the cell like this, and it will inject its nucleic acid, whether it be RNA or DNA, into the host cell. Like this is one of our body cells. Now, I say host as if it's happy to have a virus, not happy to have a virus, but it's like the virus is hijacking the cell, and then it's going to use the ribosomes in order to replicate more viral proteins like this, and it will use the nucleotides of the cell in order to replicate more nucleic acid. And so basically the cell becomes a viral piñata, and so it's got all these viruses inside. And so what I'm talking about is, this is trouble. These viruses are in our body cells, and so they're not free anymore in the fluid. And so our T cells will come along, as you can see in this picture. This is what I was attempting to draw right here. Here's a cell that is infected by all these viruses inside, and so the humoral immune system doesn't work effectively against this because they're intracellular pathogens. But this is kind of cool. And one of our own infected cells is able to take a viral particle and actually display it on the outside of the cell membrane. And that way our T lymphocyte, which is what this is, will come along and notice, let me see if I can animate this, our T lymphocyte will come along and notice, this is the T cell, an infected cell, and it will actually secrete chemical, and this is what this video is about, that will actually destroy the cell that's being invaded. It destroys it by creating pores in the cell membrane, which will allow fluid to then enter into the cell, and that will cause the cell to lysis and rupture. And so that will interfere with the viral production, or it will allow these viruses to be released where they can be destroyed in the humors by the antibodies. So this is what's known as cell-mediated, meaning that it's sort of a cell-to-cell, cell contact, cell-mediated immunity. Now, how does this look? So that's kind of the crux of the matter, but it's like the detail of it's kind of interesting. So how does the lymphocyte know the difference between self cells and non-self cells? And so if I say, here's one of our body cells, you could say that there's little proteins on the outside, like little flags saying self, like don't destroy me, it is me. And then like a germ will have a different flag on there saying, germ, destroy me, but there's no flags on the outside of the cell membrane. What we're dealing with here are proteins embedded in cell membranes, and not just the proteins, but the glycoproteins, these little tiny oligosaccharides that are emanating from the membrane of organisms. And so how do you like this? Each one of our cells in our body has specific proteins called major histocompatibility complexes, or MHC, that define our self. There isn't a single person in the entire world, this is a protein fingerprint, that has the same MHC complex that you have. And all of our cells in our body have them, that are nucleated. So interestingly enough, our red blood cells do not possess them because it's a non-nucleated cell. Now, there's two kinds of MHC markers, but I'll get into that in a moment. And so I just wanted to point out that all of our cells in our body have these major histocompatibility complexes, and they define self. And so no two individuals in the whole world will have this unless they're identical twins. And so there's a lot of effort put into identification, because that's the basis of the whole immune system. You have to be able to identify self from non-self. And so this cell-mediated response has to do with T cells, and they're called cytotoxic Ts because they're capable of destroying our own cells that are being invaded by intracellular pathogens like a virus. Sometimes they're known as killer Ts. So when an antigen-activated cytotoxic T cell, meaning that it's activated, meaning that it responds to a cell that is troublesome. So if a cell becomes cancerous, that's an indication that there's trouble, and the cytotoxic T wants to destroy that before it becomes too much of a problem and it becomes a tumor. Likewise, if the cell's infected by a virus, it would want to destroy that as well. And so this is all mediated by these class 1. There's going to be two classes. Class 1 major histocompatibility complexes. As I mentioned, all of our cells contain these class 1 molecules. And so what I was saying about them is that when a virus infects the cell, a small fragment of that virus is actually put to the outside of the surface. And so let me show you just some typical tissue right here. This happens to be epithelial tissue. Now you can't see the membrane because it's very small, but there it is right there, that line right there. But if I exaggerate it and draw, here's the major histocompatibility complexes that are on the cell membrane. So this is the MHC class 1 marker. So this is self. And so it's found on almost every nucleated cell. Okay, so I'm not going to draw every one on there, but these MHC markers are found in each one of our cells. And so this identifies self versus non-self. And so normally our white blood cells would not attack our own cells. And it's like, how could it tell? Because it has this self-identifying major histocompatibility complex. Okay, and you're like, well, all right. But here's the deal. Like if you have a cell like here, this is a typical tissue, and it has the MHC marker like this. This is the MHC class 1 class 1 marker. The thing is, when our lymphocyte comes along like this, here's the T cytotoxic cell, the killer T. I'll just say it's T sub C for cytotoxic. What's interesting about this is if I said that there was a virus inside right here replicating, and it's like, how would we know that? Well, we would know that because what's interesting is the cell that's being infected will actually, this is really clever, I find this to be most remarkable, puts a little piece of the infected protein on the outside of it, and it sort of marks it for destruction, because then the cytotoxic T cell comes along and identifies it as being abnormal in some way. I mean, this is remarkable because having a molecule attached to the protein will cause it to change shape. And so therefore that shape change in the MHC1 marker is identified by our immune system as being troubled. So an analogy that I like to use when describing this is like, sometimes, you can't tell if you're, say that this is a police car and you're driving down on patrol and you're noticing, for example, there's various stores on a busy street, and maybe this is a bank. Say this is the bank right here, and say that there's a lot of robbers inside the bank. Well, it's not necessarily good policy when there's a bank robbery in session for the tellers to be like going off and screaming or setting off an alarm that sometimes agitates the robber into shooting everyone. So what I like to think of this as being is when there's a robbery in session, sometimes the teller will push a little button and there's a silent alarm that goes off. And that's what this is. It's a silent alarm. So when the cytotoxic T cell comes along, it's like, hey, there's trouble inside the bank. And so guess what it's going to do? This is how it handles. It secretes a chemical. You ready for this? It secretes a chemical that actually creates perforations in the infected cell. So it's sort of like it throws a bomb in there and blows the whole cell up. And so this is how it handles it. It destroys its own body cell. So this is what cell-mediated immunity is about. It's a cytotoxic T cell killing your own cell. But this is a good thing. So the cell that contains a replicated virus, it carries it on the class 1 MHC marker and it exposes that foreign protein as I was describing. Let me do that again. And so like here's the cell that's being infected. Okay, so here it is, inside. But then on the outside, let me just put a little virus right there, a little virus particle. So this is what we're talking about. So the class 1 MHC marker, which is this, is exposing foreign protein, which is this. This is the foreign protein right here, like right there. And so this shows that the cell's infected or there's something abnormal about it, like maybe it's a cancerous cell. Maybe it's not infected at all, it's just a cell that's cancerous. And so it shows this to the cytotoxic T cell. And so when the cytotoxic T cell comes through the neighborhood, it's going to be able to identify it as being trouble. So it's a little marker. And so what does it do? The cytotoxic T cell then releases a chemical called perforin. And so here's the cytotoxic T cell, and it attaches to the infected cell. Here's the infected cell. And so the chemical, and I'll stay with the color here, perforin, is secreted. And so this chemical, and I like the name, perforin, because what it's doing is it's causing perforations in the cell membrane. And so the protein forms pores in the target cell. And when it does that, the cell will eventually allow fluids to come in, which it doesn't like, and so the cell will eventually lysis and then interrupt the virus from hurting us any further. And so the death of the infected cell deprives the pathogen, the virus, a place for it to reproduce. So it can't reproduce a virus unless it's inside of a host cell, because it lacks ribosomes and it lacks amino acids and it lacks nucleotides and the machinery and enzymes necessary to replicate it. And then furthermore, when these particles are then in the fluid, it exposes it in the humors to circulating antibodies, which will then bind to it and then ultimately destroy it. So These killer Ts are pretty impressive, but they destroy our own cells that are being infected, and so thus it's called cell-mediated immunity. And so here's another detail of this. Here's our infected cell. All of our cells in the body have class 1 markers, and so when the cytotoxic T cell comes along, it actually attaches to the MHC marker. This is detail, but it's kind of interesting, using a protein called a T cell receptor, but then it holds it in place with another protein, like a little hook that grabs it and anchors it there, called a CD8 protein that stands for complementary determinant 8. And that holds it in place sufficient enough so that the perforin could be released, which causes these pores to develop. And then again, as I mentioned, various solutes will go in, creating a hypertonic environment, and then water will go in through osmosis and then cause the cell to lysis, which is very effective because ultimately then the germs won't be able to replicate, as I was mentioning before. And so this interaction is enhanced by the T cell receptor and also the CD8 protein, which holds it together long enough so that the perforin is able to do its thing. And so that's cell-mediated immunity. And so there's as I was mentioning, there's a lot of class 1 proteins out there, and they can tell the difference between individuals. And so we like to think of it as one of the most polymorphic proteins known. And so that means there's a lot of genetic diversity out there in the gene pool. So there's a lot of alleles. If you're following this genetically, a lot of alleles for class 1 MHC markers. Now, I mentioned that all of our cells have this class 1. So I'll just do this. All of our body cells have this class 1 marker. But are you ready for this? There's a second type of MHC marker, and it's appropriately called, guess what? Class 2. Seems reasonable. And this is found mostly on our white blood cells. And so things like macrophages, antigen presenting cells have this class 2 marker on them, and that's going to be important as well, as we shall see. And so the MHC provides a biochemical fingerprint, I was mentioning before, that could really distinguish individuals. And so that allows for great defense, but that also means that we can't receive tissue from other humans because we're going to reject that because they have different MHC markers. And so what's interesting is I mentioned that this class 2 MHC marker, and so check this out. We have these cells, white blood cells called neutrophils, and they can be defined as being granular. In other words, they're granular sites, and they have these lobed nucleus, and they're rather common. I believe it's the most common white blood cell of them all, a neutrophil. So the likeliness of a neutrophil to encounter a germ is the most likely scenario because they're so numerous. And so how do you like this? So say a white blood cell comes along and it actually identifies a foreign organism. And it can identify it because it has foreign proteins. And these foreign proteins on the outside of a cell are referred to as the antigen. And so let me see if I can animate this. So what a phagocyte, like a neutrophil or a monocyte, is capable of doing is engulfing. Let's see if I can do this. It stretches out its plasma membrane like this, and then it takes it in. And then these two fuse together and forming like this vacuole. Ultimately, this is then broken down by the cell's lysosome, and then these viral or bacterial proteins are then used by the white blood cell, okay? So the white blood cell then places them on the outside of the cell. So this is kind of similar to what I was talking about before, but an infected cell, but this is different. This is a white blood cell. So this is a white blood cell. And remember, white blood cells have a MHC class 2 marker. And so what they do is they put these little pieces of bacteria, protein, on the outside of their cell. And so it's sort of like a hammock. So they put it on the outside of the surface. And what they're trying to do here is present the antigen. And so the white blood cell that's capable of doing this is sometimes known as an antigen presenting cell. And it's like, well, what is the story with this? Well, if the germ itself is encountered by a B cell, then that B cell can become activated and secrete antibodies and then ultimately destroy it. But here's the thing. The B cells are found in very few numbers because of their great diversity. But here's the thing also. A T cell could come along and identify it as well. But again, those numbers are few and far between. So if a white blood cell, like an antigen presenting cell, comes along and presents this, then a B cell may not need to identify the foreign antigen. It could actually come along and identify an antigen presenting cell and then realize that there's a germ in the body. Likewise, another kind of T cell, let me introduce you to this. Another kind of T cell, really cool, is called the helper T. Sometimes a lymphocyte called a helper T will encounter an antigen presenting cell. And when it does that, it is then able to get excited, it's activated, and it realizes that there's trouble in the body, there's a germ in the body. It will divide and become more numerous. But what it does is it will secrete chemicals in the fluids that will actually call B cells to the area and cytotoxic T cells to the area to come destroy the germ. And so these helper Ts can communicate by secreting chemicals. Sometimes those chemicals are called lymphokines because they're calling other lymphocytes. Sometimes they're called chemokines because they're chemicals that will cause movement. And the particular name of them are called interleukins. And so this is what a T cell can do. And so T cells are then alerted to an infectious agent either by encountering it themselves or by encountering an antigen presenting cell. And so these T helper cells are rather important because they can amplify the fact that there's a germ. And so they're kind of, if I can use this analogy, it's very difficult for our defense systems, like for example in countries, to look out for every possible terrorist, every possible criminal. So the best way to defend against trouble is not necessarily, it'd be great if there's a criminal and a police happens to notice that they're present, like in an airport. But the best defense of all is to have all of these helpers. And these helpers will notice when there's a trouble, and they'll actually call for the real police to come to an area. And that's what a T helper cell does, is it calls for other lymphocytes to come into the area. And so a T helper cell can bump into an antigen presenting cell, and it has receptors to bind to that. It has a receptor that binds to a class 2 MHC marker, and it knows that there's trouble because there's a little protein here. And so it starts to produce chemicals that will call other lymphocytes to the area. So thus it's called a T helper cell because it's very helpful. And so when you think about it, T cells can be sort of characterized into two main kinds. There's the cytotoxic T that's going to respond and destroy our own cells that are that have a displayed antigen on the MHC1 marker. And then a T helper cell will specifically bind to other white blood cells that are also infected. So in other words, an antigen presenting cell. And so when an antigen presenting cell and a T helper cell come together, what's going to happen is the T helper cell is then going to release chemicals that will call other lymphocytes, whether they be B or T, to the area. And then when the cytotoxic T cell comes, it'll come a calling, and then, as I mentioned before, it'll attach to the infected cell, secrete its perforin, and destroy the infected cell. And so these helper Ts are really, really important. And so, again, sort of a review of this. Now, this can happen in a variety of different ways. So, if a macrophage attacks a pathogen, as you can see here in this orange with the purple dots, it'll break up these purple dots and display it. So this is an antigen-presenting cell. So this is an antigen-presenting cell, and it's going to be on a class, that's right, class 2 marker. And so what can happen is the antigen MHC receptor on the helper T cell will identify that there's trouble. And so it'll identify that. Now, it'll either identify it or the MHC itself will secrete chemicals calling the helper T. I didn't mention that. But then this will eventually divide the helper T, and this will produce chemicals that will call in other B and T cells. Or, so that's a helper T. This is the T helper. Or what can happen is just an ordinary, it says virgin, but an ordinary cytotoxic T could bump into an antigen-presenting cell if it's never encountered it before. And then what will happen? The cells will divide and divide and divide, and then eventually they'll be in larger numbers and then ultimately destroy infected cells. That's what cytotoxic T cells do. Or it might encounter, if you've been sick by that germ before, it might encounter a memory T cell. And in that case, there's a lot more of them, and the response is more rapid, and therefore you don't get sick. So cytotoxic T cells activated either by a class 2 marker right here, or it's activated by a class 1 right here. And so as it turns out, the chemical that's secreted by the helper T, this chemical right here that's calling other cells, is called Interleukin 2. And so this is what I want to take this conversation. It's getting a little detailed, but just a little review, just to bring everybody onto the same page. So a cytotoxic T cell can destroy our own cells, and a helper T can attach to an antigen-presenting cell, realize that there's trouble, and then communicate that with other lymphocytes. And so this is what I want to talk about. Like, what are these chemicals that are able to do that? Well, the chemicals are called cytokines or lymphokines or interleukin. But in so doing, the T helper cell actually can be responsible for calling, using these chemicals by calling in B cells. And when it does that, it creates a lot of antibodies, so that helps in humoral response, and it also calls the T cytotoxic cells, which are responsible in the cell-mediated immunity. So this is kind of the linchpin, in other words, the key to our whole immune system, because it really, now you're like, well, do you really need it? Yes. Well, because B cells can find the germ on their own. Yeah. They could find the germ on their own. Cytotoxic T cells can find germs on their own or encounter an infected cell. But it's more likely that they're called to the crime scene by the helper T cell. Why? Because it's more likely that the antigen-presenting cell will encounter a helper cell and get this whole process going. And so both types of immune responses, both humoral and cell-mediated, occur really through the help of antigen-presenting cells and helper T cells. And so that's really important. So the antigen-presenting cell via the helper T cell is really the heart of both types of immune systems. And so they help ultimately to call the effector cells. The effector cells are the cells that are affecting a response. In other words, the plasma cells that are secreting the antibodies and the cytotoxic T cells that are actually destroying infected cells. So here it is. Here's the antigen-presenting cell. Here's the helper T. So when the helper T bumps into this, it's like there's trouble. And so what happens is that the helper T will divide, and that's what it means to be activated, where it needs to increase in number. And so sometimes the helper T encounters it, physically bumps into the antigen presenting cell. Sometimes the helper T cell is called and is activated by interleukin 1. And so this chemical will actually stimulate the cell to divide. And then this chemical, which is I was drawing before as being like sort of like red sound waves coming out like this, these chemicals are called interleukin 2. Interleukin 2. Detail, but it's kind of cool. The helper T cell secretes interleukin 2, which calls and stimulates the cytotoxic T cells and the B cells to come to the crime scene. Interestingly enough, the chemical also causes it to self-divide as well. And so what we have going here is, in terms of a linear progression, is the antigen presenting cell encounters a bacterium. It puts the molecules onto its MHC2. The helper T identifies it by attaching with a complement determinant 4 receptor protein, holds it in place, allowing the interleukin 1 to stimulate the T cell to divide, then allowing the T cell to secrete interleukin 2, which causes B cells and T cells to come into the area, which are very important, because once those cells are called, then the immune system can work most effectively. And so the T helper cell is really important in both the humoral and cell-mediated immunity. And it does that by cell signaling. And that's what I mean by cell signaling, is that it secretes interleukins. So in other words, the helper T cell, it's kind of a funny cartoon, the T cell with the helper hat on it. So this is what a helper T really looks like. It secretes interleukin, which then causes B cells and cytotoxic T cells to come to an area. And so in conclusion, the whole immune system can be summarized by this very useful diagram. Over on the right side, you can see it's cell-mediated immunity. And so this is the cytotoxic T cell. So it's either going to encounter an infected cell, one of our own cells, and destroy it. And then when it gets all activated like that, it's going to divide, and produce many clones of it. And then ultimately that will remain, once the germs are destroyed, there'll be a lot of memory cells. And so we'll be able to respond better in the future. Likewise, a B cell can, fortunately, there's many different kinds of B cells, and they all have these different receptor proteins. But if the right one happens to encounter the antigen, that's going to cause the cell to divide and clone, so many copies of that cell through mitosis, forming effector cells. And these effector cells become really large and secrete copious amounts of antibodies. And so when they do that, they're called plasma secreting cells. And they secrete these immunoglobulins called antibodies, and they go out and dominate in the fluids and destroy germs. So that's humoral response. And you're like, well, what's this in the center? Well, the more likely scenario is that a germ comes in and it's encountered just sort of like our nonspecific immune response. A macrophage comes and eats it and presents it. It's an antigen presenting cell. And then, a helper T cell encounters that, which causes the helper T cell to become activated. And when it becomes activated through interleukin 1, it then secretes interleukin 2, which then stimulates cytotoxic T cells and B cells to come to the area. They become effector cells, and then everything is enhanced. Now, ultimately, the antibodies, the concentration becomes so large, and it just rains down terror on the antigen, and it's destroyed. And you're like, yes, that is how my immune system works. Yes. But then this is the best part. Save the best for last. The whole immune system is based on immunity. So check it out. When the antigen shows up again, the second time it shows up again, you're not going to become sick, because we have these memory B cells. Because after the first war was over, a lot of the cells remain as memory cells, and so they're able to respond very quickly. So both memory T cells and memory B cells respond so rapidly to the second exposure of the same antigen, we don't become sick, and thus we're immune. There you have it. So hope you enjoyed this video on cell-mediated immunity, and thanks for watching.